People may want to move back one row. So we're going to talk about, and I have to say, I'm using the same slides I used for a talk at a meeting in Yale 3 days ago. So you're just getting their slides. And so anyway, so finally, an established mechanism for health impacts of electromagnetic fields, how diverse health effects are produced by what are called downstream effects. And we'll explain what that means in a little bit. Okay, so let's see. This thing doesn't seem to be working.
You have to press escape to get it out of that escape key mode.
Oh, okay.
There you go.
Okay, thank you. All right, so, and I'm going to start with some ancient history. And it's ancient history that the industry has totally and utterly forgotten, despite how important it is. And that is, back in 1971, the U.S. Office of Naval Medical Research issued a report listing various types of health effects produced by non-thermal exposures to electromagnetic fields, okay? So these are not effects that are produced by heating. These are produced under conditions where there's essentially no heating. And in this, they list quite a number of things, including 40 different, what might be called neuropsychiatric effects. So these include changes in the structure and function of the brain. These include changes in various types of psychological responses and changes in behavior, okay? And I'll talk about some of the specifics a little bit later. Now, they include 8 different endocrine effects, and so various kinds of hormone changes. They included cardiac effects, influencing the electrical control of the heart. So these included changes in the electrocardiogram. This looks at the electrical activity in the heart. They include arrhythmias. They include tachycardia, rapid heartbeat. They include bradycardia, slow heartbeat. And more recently now, we now see also heart palpitations. So lots of things that have to do with the way in which the electrical, the heart activity is controlled electrically. Okay, so chromosome breaks and other changes in chromosome structure. So these are in the cells. The chromosomes break, and they show, when they break, they can rearrange and produce new kinds of structures. Histological changes in the testis. So the testis is very sensitive to the EMFs, and you get changes in the structure of the individual cells and how they fit together in the testis as a function of these non-thermal exposures. And you get increases in cell death, what we now call apoptosis. That term wasn't known 46 years ago, but but we certainly know it now. And this is a process that's very important in neurodegenerative diseases. You get a lot of this cell death. And the Naval report also provided over 1,000 citations, that is individual studies, that documented all of these various non-thermal effects, okay? So there was already a huge amount of evidence 46 years ago on this. And you tell industry this, and they'll probably have a, probably go into seizures or something. Anyway, now, following 1971, there have been dozens of reviews. In fact, I listed over 10 dozen reviews in which the reviewers, so these are published reviews in which the reviewers looked at anywhere from a dozen to over 1,000 different studies that clearly show that there are non-thermal effects of various sorts. And so there's a large amount of the non-thermal effects. And among the effects that have been repeatedly documented since the Office of Naval Medical Research came out was we have lowered male fertility, including lowered sperm quality and function, and also lowered female fertility. Female fertility is less studied because it's harder to study, but it still is impacted. And there are also increased levels of spontaneous abortion. And so these have been studied in experimental animals, and they've been studied in people. So let me just say, you know, it's known that men all over the world have had declining levels of sperm numbers and also sperm quality with increasing time. Those are unexplained, but we know the EMFs, in fact, can cause these. And so that's sort of an obvious causation that we know about. You get oxidative stress, and oxidative stress is known to be involved in many, many different chronic diseases. You get damage to the DNA of the cells, okay, and they occur of 3 different types. And so you get single-strand breaks in the cellular DNA, you get double-strand breaks in the cellular DNA, and you also get oxidized bases. And the one I've listed here is 8-hydroxydeoxyguanosine. Probably doesn't mean much to you, but anyway, it's a thing in your DNA which gets oxidized. And when it gets oxidized, you know, it can produce mutations at the cellular level or at the organismic level if it's passed on. And the same thing is true of the other changes as well. They can also produce mutations, but of different sorts. So cancer is likely to be, so we know there's cancer increases. Those are likely to involve DNA changes that was just discussed there. But there's also evidence for increased rates of tumor promotion-like events. So tumor promotion is another part of the process that produces cancer, and it's elevated as well. It's been shown to be elevated in certain studies, and in response to these electromagnetic fields. There are widespread neuropsychiatric effects, including depression, and I'll talk about a number of those later on. There are therapeutic effects. Okay, so there are genuine therapeutic effects of these EMFs. And so everything is not damaging, but most of the things that I've listed obviously are. But it's important to acknowledge that these therapeutic effects are real, that they are non-thermal, they don't have anything to do with heating, and that they also have to be explained in terms of the mechanism that's occurring. So cataract formation. It was previously claimed that cataracts were due to heating, were thermal. We know now that they're not due to thermal effects, and in fact, they can be produced by the mechanism I'm going to discuss later on in my talk, along with everything else. Breakdown of the blood-brain barrier is another thing that occurs. And so the blood-brain barrier is something that protects the brain from toxic chemicals and from infectious agents. And so when you get a breakdown of the blood-brain barrier, that's a serious issue. And we know this breaks down in response to these electromagnetic field exposures. Melatonin depletion. So people at night normally have high levels of melatonin, helps them sleep. On EMF exposures, the melatonin levels are very low at night, and you get sleep disruption, not surprisingly. So, so there's all these things. Now, despite all of the evidence for all these non-thermal effects, our current safety guidelines are based on the assumption that only thermal effects need be of concern. So basically, the situation is that you know, we've been using this assumption of only non-thermal effects for something like 30 years, and we knew all along that that was wrong, or we should have known all along for paying attention to the evidence, and yet this is still going on.
Dr. Paul?
Yeah.
Since we have a small group, are you open to questions, or do you want
Yeah, that's all right. Go ahead.
I think probably a lot of us have heard a lot of these types of things before.
Yeah.
And the burning question in my mind is, since Cecilia was talking about how we're bathed in this.
We don't. Why don't we?
Sure.
Yeah. Go ahead.
We got to come a little closer.
Oh, sure.
Yeah, you're gonna. Right. Okay. Okay. There you go.
Since we're bathed in this, and I now have a meter also, so I found it very, very helpful to know where what is.
Yeah.
How is everybody not a lot sicker than we are or appear to be?
We're getting there. That's the problem. Let me just say, the effects, most of the effects that have been studied, start out slow, and at least some of the effects, when they're relatively modest, are reversible. But as we have longer and longer times of exposure, the effects get more and more severe, and they become irreversible. So, you know, so we're not aware a lot of times about these things. And so, for example, you know, I'll tell you later, I mean, many of the neuropsychiatric effects that are produced are the same things that people complain about all the time. Okay? I can't sleep, I'm tired, I can't concentrate, I'm depressed, I have anxiety, you know, all these kinds of things which people complain about all the time, all over the place. We know all those things and a number of others are caused by these EMFs, okay? And so, you know, so lots of times we're not doing well, but we don't understand what the reason is. And, yeah. Mm-hmm. So anyway, so I hope that answers your question. Okay, let me put this back. So we have these current safety guidelines that don't make sense with the science. And in 2015, there was an appeal made to the United Nations and all the member states that was signed by 225 scientists from 41 countries. I was one of them. I take no credit for this, by the way. I wasn't leading this effort. And the appeal stated that the current safety guidelines and standards are inadequate because they don't take into consideration non-thermal effects. And so these 225 scientists, signers, had each published peer-reviewed scientific papers in this area. Okay, so they're not just people picked off the streets. These are all people with high levels of competence who are published peer-reviewed studies in this area. And for a total of 2,000 such papers. So this is a very substantial amount of the total literature in this area. And these 225 scientists say the safety guidelines are no good because they don't take into consideration non-thermal effects. So, and let me say there have been a number of other statements that have been made. I'm sort of focusing on this one because I think it's the most important of them, but it's certainly not the only one. They go up, I don't know, I think we're up to somewhere close to 20 different ones that I know about. So, so there should be no question that there is an international scientific consensus among independent scientists, okay? I'm saying independent, that is those that are not closely tied to the industry, that there are quite a number of non-thermal effects and that the safety guidelines are inadequate. Okay, so now, how are the non-thermal effects produced? Okay, so we know they exist, but that doesn't tell us anything about how they're produced. And I have to say, this has been a real challenge. And the industry has claimed that they could not, these were impossible because they claimed the physics told us it was impossible, okay? And I take the physics seriously, having gotten a degree in physics. And as you'll see, the situation now is that the physics and the biology are pointing in exactly the same direction. There's no conflict there at all. Okay, so, but let's go on to the question of how does this system work? So I first found two studies published in the 1970s that suggested how it worked, and then I found a few more, and then going up to 23 and 24 and then 26 studies, all of which pointed to the same mechanism. And now, in fact, I know there are more than that, but I haven't published on the others, so we'll leave them out for the time being. Okay, so, so what is it? And so the papers I've published, the first one here that you can see is, you know, so electromagnetic fields act via activation of voltage-gated calcium channels. That's a real mouthful. It may not mean much to you, and I'll tell you how this thing works in a minute, to produce both beneficial, that is the therapeutic effects, and adverse effects, which is all the other things that I've talked about here. So that paper, by the way, was honored by being placed on the Global Medical Discovery website as one of the top medical papers of 2013. And since it came out, which is fairly recent here, it's been cited by other authors. Well, I guess not all other authors, because I cited it a few times myself. It's been cited 124 times now, according to the Google Scholar database. So it's attracting a lot of attention. Now, and that's important. And this was my first paper on it, so it's not like I had a big reputation already to bring people to it. So anyway, so there have been a number of other studies. This one was that I published, one on therapeutic effects here, and another one that's a very complex one that I'll talk about that has a lot of things in there, including why these voltage-gated calcium channels are so sensitive to the EMFs. And then there's another one on neuropsychiatric effects. And then in 2015, and this was a paper that was aimed at the industry, telling the industry what they should do. I wish I could tell you they ran out and did everything, but I guess I can't tell you that. And then there's finally a paper that shows that the way EMFs work in plants is actually very, very similar to the way they work in animal cells, including the cells of our bodies. So this is a very, very general mechanism that we're talking about, okay? So, how then are the non-thermal effects produced? As I said, there are 26 studies that showed that the EMF effects could be blocked or greatly lowered by calcium channel blockers. So calcium channel blockers are drugs that specifically block these voltage-gated calcium channels, okay? And so what this shows is you have to activate those voltage-gated calcium channels to get the various responses that were being studied, okay? And when these studies looked at more than one response, so, you know, 1, 2, 3, 4 different responses, what they found is, you know, if one of them's blocked or greatly lowered, they're all blocked or greatly lowered. And what does that tell you? What that tells you is that there are many responses that are blocked or greatly lowered, not just a few. So we're not just picking out a few things that happen to go through this pathway, but lots of things go through this pathway. And so that's important, obviously. And so when these studies were done, they used 5 different calcium channel blockers. Why is that? And which had different structures and bind to different channels and different sites on the channels. And the only thing in common that they have is they're calcium channel blockers. It block the VGCCs, the voltage-gated calcium channels. I haven't introduced that term. VGCCs is an abbreviation for voltage-gated calcium channels. So you hear me say that a lot. And so what that tells you then is, okay, these things are each thought to be highly specific. So it's very unlikely that even one of them would have a different mechanism. To have all 5 producing these blockages really argues this is the mechanism that's involved. The voltage-gated calcium channels, the VGCCs, are involved. Now, okay, so now, okay, so let's, I haven't said anything about the VGCCs, where they are, what they do, what happens when you activate them, right? But that's obviously important here, so what can we say about that? Okay, so first of all cells in our bodies have a membrane around the cell. It's called the plasma membrane, or sometimes called the outer membrane. And the VGCCs are in the plasma membrane. They're in that membrane that surrounds the cells, okay? Such that, because they are calcium channels, when they open up, so when those VGCCs open up, they allow calcium to flow into the cell, and it's the excess calcium in the cell that's responsible for most, if not all, of the biological effects. Okay? Now, So how can you relate to that? Well, we'll tell you a lot of the important things the VGCCs do and the calcium does. But under normal circumstances, the calcium in the cell is about 1/10,000th of the calcium outside the cell. And calcium ions have two charges, and it turns out those charges also tend to drive the calcium into the cell. So there's what's called an electrochemical gradient of calcium, something like a millionfold. Okay, so there's a big force that's driving calcium into the cell, and normally the cells, the cells keep the calcium levels very low. And that's obviously important. Okay, now it's also true that that you do get rises in calcium in the cell. But they're usually, just for short time periods, and then the calcium is chucked out of the cell. So calcium signaling is very important, and so you do get short-term things. But when you activate these channels, it causes major problems because you don't want too much calcium in there, or you're going to screw up a lot. Okay, so let's go on. Okay. Now, there are 5 further types of evidence then that each provide further support for the VGCC mechanism for non-thermal effects. Now, before I go into this, I want to emphasize one thing about this. I'm going to be focusing on microwave frequency EMFs, okay, right? And the reason I'm focusing on that is because our exposures to them keep skyrocketing, right? So that's a big thing. But it's also true that the extremely low frequency EMFs, such as what we get from our power wiring. We've got, in the US, we have 60 hertz power wiring. In Europe, they have 50 hertz power wiring. Those effects also go through VGCC activation. Same target. I'm not saying they're equally active, same target. There's some studies on intermediate frequencies that they also go through that. There are even studies that static electrical fields and static magnetic fields also go through VGCC activation. The magnetic fields are particularly interesting because of the physics, and you're a physicist, right? Are there any other physicists here? No. Okay. Well, the physics is very interesting here, so I want to deal with that when I can. Okay. So.
One quick question.
Yeah.
You mentioned the therapeutic effects.
Here, you want to try this again? Yeah.
I can just talk loud.
No. Yeah. Go ahead.
You had talked about the therapeutic effects before.
Yeah.
So if we're getting it sounds like we're getting force-fed calcium into our cells. Mm-hmm. So what's the opposite of that could be therapeutic?
I'll talk about that. Okay.
All right.
Yeah. In general, the therapeutic effects come from relatively modest activation of the VGCCs. But they come through the same mechanism, and I'll talk about how you get those. Okay, so so the VGCCs and some other voltage-gated ion channels have a structure in them which causes them to be opened in response to electrical changes. Okay, so the plasma membrane that I talked about before normally has an electrical charge across it, okay? And when that charge across the membrane changes, it can activate these VGCCs in the normal physiology, in the normal, in our normal cells under normal conditions. No EMFs involved at all. Okay, they can be activated, and so they have this voltage sensor, which is extraordinarily sensitive to the electrical effects of the EMFs, and I'll show you why it is shortly. And for that reason, I've argued that the voltage sensor is the primary, possibly the sole target of the low-intensity EMFs. That's the structure. Okay. Now industry acknowledges that microwave and lower frequency EMFs put forces on positively or negatively charged groups. So you have a chemical that's got a plus charge or a minus charge on it. There are forces that are put on it. And in fact, when you cook food in a microwave oven, that's the way it works. Okay, so basically, you put your food in a microwave oven, and the microwave oven basically joggles these charges back and forth. That in turn heats the food, and that's what cooks it. Okay, so then that's been known basically since the time of World War II. So industry acknowledges that microwaves and lower frequency EMFs do put forces on charge groups, but they state that the forces produced by these low intensity EMFs, the non-thermal ones, are too low to produce biological effects. Okay? And so we need to consider that issue. Okay? How can these very low intensity EMFs produce biological effects, and specifically, how can they activate the VGCCs? Okay? That's it. Okay, so if you look at this figure, and you'll see, you see these cylinders in the figure? Some of them are sort of a blue-gray color, and then there's one that's orange. It's got a bunch of pluses on it. You see that? And if you look at that, you'll see, if you look at, there are Roman numerals up there, 1, 2, 3, and 4. Those are very similar, what are called domains. They're parts, structural parts of this, the VGCC protein that are repeated. Okay, so there's almost identical domain 1, another one domain 2, another one domain 3, another one domain 4. Okay, now, and the voltage sensor is that orange colored, and it's just orange so you can look at it and say it's different, okay? The orange colored cylinder is the one that has the charges on it, and those charges are the voltage sensor, okay? So you got 5 charges on the one on the left, in Roman numeral 1, and then 5 more on 2, and 5 more on 3, and 5 more on 4. A total of 20 charges, okay? That's the voltage sensor, okay? Now, what are those cylinders? Those cylinders are what are called alpha helices, which is a structure in the protein which kind of goes around like that to form kind of a cylindrical structure, okay? And so the charges are on one of the 6 alpha helices. It's actually the 4th helix in each of these, okay? And what happens when you activate the channel is basically that those charge groups kind of pull out from that structure and go out into the extracellular fluid, okay? So the extracellular fluid is the stuff that's on the top. So basically pulled up. And so all 4 of those have to pull up, and then it'll open the channel, whoosh, lots of calcium, okay? And okay, so this figure was taken from a paper that was published by Professor Anna Dolphin from the UK, and it's a very nice figure. But there are a number of things about it that are misleading when you look at the actual protein, okay? The reason I'm using this figure is it's easier to understand than the real thing. When you look at that, it's very difficult to understand. Okay, so there are a couple of things I just wanted to point out, and that is, instead of being planar, instead of being just stuck out, 1, 2, 3, and 4, these are actually wrapped around each other in kind of a cylinder, okay? And the channel is actually in the middle, okay? So they're wrapped around each other, and the other thing which is important is these alpha helices are actually tilted. They're not straight up and down, they're tilted, okay? Including the alpha helix that's got the charges on it that's in the voltage sensors. All of those are tilted. Now, why is that important? It's important basically because all of those charges are all within the fatty section of the lipids. Okay, so if you look at that, well, you can't really see it very well. You look at the little circles going across the top and the bottom of the membrane. In between are fatty acid chains. So they're kind of a fatty, oily kind of structure, okay, in there. And those charges are all within that region, okay? They've got protein around them, but they don't have any, they don't have any water there. Now, why is that important? Well, there's something called Coulomb's law, which is written there, and it's expressed in Gaussian units. That probably isn't important to you. But the important thing here is that the forces on charged groups, according to Coulomb's law, is inversely proportional to the dielectric constant of the medium they're in. Okay? Now, the dielectric constant of the fatty part of the membrane is about 1/120th of the dielectric constant of the watery parts of the cell. Okay? So what does that mean? It means the forces are 120 times stronger on each of those charges than they would be if they were out in the aqueous phase in the watery parts of the cell. Almost all the charges in the cell are out in contact with the watery parts. These are not. That makes them very different, and that makes the forces on them much stronger. Okay? Now, so we're getting to this question of how can this work with the physics? How can this work with very low intensity EMFs? That's what we're getting to. Okay, so this is a picture of or a painting of Coulomb, who proposed this. I used this slide in Paris a couple of months ago, so it's in French, and he proposed this in 1784, just before the French Revolution. So it's been a long time. This is old physics, and so when you compare the force on the voltage sensor with the force on singly charged groups that are elsewhere in the cell, namely in the watery parts of the cell, the force on the voltage sensor is 20 times 120 times 3,000. Where's the 3,000 come from? I haven't said anything about that, right? It turns out that because the plasma membrane has a very high electrical resistance, whereas the watery parts of the cell have very high electrical conductivities, the electrical forces are very highly amplified right across the plasma membrane. And it's been estimated that's about a 3,000-fold amplification. Okay? So the forces on these charges then are 20 times 120 times 3,000. These are approximate numbers. I'm not saying that they're precise. Are approximately 7.2 million times stronger than the forces on singly charged groups in the watery parts of the cell. What does that tell you? It tells you the industry calculations are off by 7.2 million, or roughly 7.2 million. All right, well, that's enough to throw the whole industry claims in the trash, and that's exactly where they belong. So the other thing I think that's worth pointing out is that heating, which is the basis of the current safety guidelines, is produced mainly by the forces on singly charged groups in the watery parts of the cell, okay, in the aqueous phases. And so what that says then is the safety guidelines are allowing us to be exposed to EMFs that are approximately, this is not a precise number, approximately 7.2 million times too high. Okay, that kind of blows the mind, doesn't it? Okay, so this is, this is really quite striking. Okay, so, oh, here we go, sorry. Okay, so we have a situation now where the physics and the biology are pointing to exactly the same answer, okay? The biology says these VGCCs are the main target. The physics tells us why it's the main target. It's because the voltage sensor that controls it is so sensitive to the electrical forces. And so everything's coming together, everything's pointing in the same direction. That's important. Okay. Now, here we have, okay, so how does the whole system work? And let me just say, I know a lot of this stuff's going over your head, okay? Don't worry about it. You will get certain things that will not go over your head, and that's the thing you want to hold on to, okay? Now, so we have microwave and lower frequency EMFs over there on the left activating the VGCCs. And so they open up and you get increases in intracellular calcium. Intracellular calcium normally designated Ca2 plus I. And so that Ca2 plus I there it represents intracellular calcium. And so we're going to talk about things going over to the right from Ca2 plus I, okay? So one of the things that happens when you increase intracellular calcium is you get a big increase in nitric oxide. Nitric oxide is, the structure of nitric oxide is NO, so that's what that NO is. And the reason you get that is because two of the enzymes that make nitric oxide are calcium dependent. So when you have very low levels of calcium in the cell, those enzymes are essentially completely inactive. But when you get a big increase, you get a big increase in nitric oxide production. So that can go. And then nitric oxide can act along two pathways. And one of them is what's called the nitric oxide signaling pathway, which is the one that goes just right off to the right from nitric oxide. So you get a big increase in cyclic GMP, and that in turn activates something called a protein kinase, which responds to cyclic GMP. It's called a G kinase. And that produces therapeutic effects. Okay? And the therapeutic effects, I argued this in my first paper on EMFs. And interestingly, Arthur Pilla, who's a real expert on therapy, he's been working on it for over 40 years, came to a very similar conclusion in a paper that was published in almost identical time. Okay? That's the way the therapy works. And so there are real therapeutic effects, and that's the way it works. Now, you have also, so calcium can also increase something called superoxide. That's just the next arrow down from calcium going off to the right. And superoxide can react with nitric oxide there to form peroxynitrite. And the structure of peroxynitrite is ONOO minus. So that's peroxynitrite down below. Are you following me okay? Okay. And peroxynitrite is a potent oxidant. It's not a free radical, but it breaks down to form free radicals. And so and those free radicals are highly reactive. So you go from peroxynitrite to the free radicals just to the right of it. And then they produce oxidative and nitrosative stress, so they can produce a lot of damage in the cell. And all of those things, the peroxynitrite, the free radicals, the oxidative stress, are involved in producing pathophysiological effects, so disease-causing effects, things that damage our bodies. Okay. That's not the whole story about the pathophysiological effects. There's another part of it that involves excessive calcium signaling. Okay, so calcium signaling is very important, but you don't want too much of it. And obviously, when you let lots of calcium flow in the cell, you are going to get too much of it. And so you can get pathophysiological effects that involve excessive calcium signaling. Okay? So that's basically the overall understanding of how the system works to produce both a variety of pathophysiological effects and also the therapeutic effects as well. Okay? And so, do you have another question?
Sorry, one more.
Use the mic. Okay, go ahead.
So the action here is right at superoxide. When it makes that Y.
Why does it do it? It does it. I mean, the calcium does it. I mean, excess calcium does it.
It's going one way or the other way, depending on superoxide, that you were saying?
You mean, why do you, okay.
Between the therapy
and the peroxynitrite pathway. Yeah, okay, that's a very good question.
Well, my question is,
yeah.
Is sulfur involved in that? Stephanie Seneff from MIT.
I know. I don't think it's true. Okay.
Okay.
All right. So let's not worry about that. Okay. Okay. So, all right. So I mean, it's a very good question. Why would you get predominantly the therapy pathway under some conditions, and in other conditions you get the peroxynitrite pathway? Okay. And I think there are sort of two answers that are both important here. One is that, in general, the therapy pathway tends to be the main one when you have a relatively low amount of calcium in the cell, and you don't get that much superoxide produced. Okay, under those conditions. You do get some, but you don't get a huge amount. And so when you get higher amounts, you get peroxynitrite, and then you tend to have that pathway. The other thing, and this is not at all obvious from this figure, okay, but what's true is that each of those pathways inhibits the other. Okay, so when you've got one pathway turned on, it tends to lower the other. You got the other pathway, it tends to lower the first. Okay, and I've published on that, but not in a place that you're going to find it easily. Okay, so but anyway, it's true. And I don't want to get into gory details on this, but that's something else that's important here, that they tend, at least to some extent, not be both they tend not to turn both of those on at once, because each of them tends to inhibit the other. Okay, so okay, and then finally you've got the calcium signaling, which is important. Okay, now we've talked about a lot of things that are going on in response to these effects, and you can't see that all that well. And what I want to do is talk about one specific thing that's going on, because I think it gives a lot of information about things that are very surprising and also very important. Okay? So let me just say, I mentioned before that you get single-strand breaks in the cellular DNA, and the way in which you look at them is through this thing, and I don't know that you can see this as well as I'd like. What's called a comet assay. You see on the top of that, it says comet assay. And the rest of this slide, at least some of the rest of the slide's in German, okay? It's because it's a group that was done this in German. So this was published by Lutz and Adelkofer, and I spelled Adelkofer's name wrong. It doesn't have an E in it. It only has one E, and the first E shouldn't be there. And it's entitled Objections Against Current Limits for Microwave Radiation. So what are we looking at here? We're looking at, first of all, you have DNA in these cells, and normally DNA is a huge, long, gigantic, long molecule, okay? Incredibly long molecule, okay? If you get lots of single-strand breaks, they're little short pieces. And if you put these in alkali, which is the way this is done, the DNA, the double strands of DNA break up into single strands, and the little single strand pieces can move. And if you then put them in an electric field, and DNA is negatively charged because it's got all these phosphates on it, it'll move towards a positively charged pole, okay? So I don't think you can see these very well, but if you stretch your imagination, you can see that there is a tail in the one that says gamma radiation there, and then another one that says RF EMF, radio frequency EMF, and it's going off to the right. Can you see that? Yeah. Okay. Your eyes may be better than mine. Anyway, so there's a tail going off, and the size of that tail tells you basically how many single-strand breaks you got. Okay, and so what this, you can actually see it better on this computer here than you can see it on the screen there. I don't know if, yeah, you can go, if you look at it. So what this, and the way they talk about this now is that gamma radiation, the equivalent of 1,600 chest x-rays, so a lot of x-rays, okay, produces about the same amount of DNA damage as what they call 24 hours on a mobile phone or cell phone. So here we have a situation where you've got a cell phone with a tiny little battery producing roughly the same amount of DNA damage as a very high-powered X-ray machine running with 1,600 chest X-rays. Okay? So that's really stunning. Okay. Now it turns out that this study actually underestimates the effects of real cell phones. And why do I say that? Well, if you look at the figure on the bottom of those three, what do you see? You see 24 hours continuous wave. Okay? You see that? See that on the bottom? Yeah, okay. So now real cell phones don't have a continuous wave. Real cell phones pulse up and down, okay? And we know from other studies that pulsed EMFs, things that pulse up and down, are much more biologically active than things that just have this continuous wave, you know, that just go on, okay? So this, in fact, greatly underestimates it, and I'll show you that that's true in a little bit, that you get these single-strand breaks. Okay, so is there evidence on that? And of course, I wouldn't be telling you about it if there were not. So there were a couple of studies done by the same research group in collaboration with another research group of Rudingers. Adelkoffers in Germany, Rudingers in Austria, and they published another paper basically showing that, that when you have pulses, you get more DNA breaks. And then they set up in the bottom one, in the Schwartz et al. Article, they set up pulses that very closely simulate the actual pulses you get from real cell phones. Okay? And so they tell you about how they did that it maximizes both the megahertz and sub-kilohertz power variation. So it pulses in both ranges. So these are very similar pulses to what you get with a real cell phone. And what do you find? You get still more DNA breaks even at very low intensities. Okay? So now we're talking about real cell phones being vastly more active than the 1600 chest X-rays. And so what does this say? This says that the industry propaganda has been claiming, oh, ionizing radiation is dangerous, got to worry about that. Non-ionizing radiation, you don't have to worry about it at all, okay? And yet, now what this looks like is non-ionizing radiation can be much more dangerous at the same level of energy as ionizing radiation. Okay, so that, though, raises another question, doesn't it? How can this happen? What's the mechanism? How do you get all these DNA strand breaks from this, low amount of, cell phone radiation, right? That's another question. And so what's my answer to it? My answer is that both the microwave frequency EMFs and the ionizing radiation work to produce the DNA strand breaks via free radicals. Okay? They both produce free radicals. When you get the free radicals, if you go and you look at this figure that we just looked at, what you find is there's 3 steps where you see a high level of amplification. When you've got 3 high levels of amplification, you can get a gigantic response starting with a very small amount of energy putting into it, okay? And I think that's exactly what's happening here. So first of all, when the VGCCs are activated, and they open up, you get about 1 million calcium ions flowing in the cell per second. And you can have lots of these in a cell, and you can get huge amounts of calcium going in. Okay, so that's the first thing. First level of amplification, something like 1 million calcium ions flowing in per second, that it's open. Then you have the calcium acting basically catalytically to increase nitric oxide production and increase superoxide production. So that's the second level of amplification. And then there's a third level. Why? Because the production of peroxynitrite there is proportional to the product of the concentration of nitric oxide times the concentration of superoxide. So there's a third level of amplification there because it's a product effect, okay? So 3 levels of amplification, and what do? You get lots and lots of free radicals off a small amount of microwaves. So now ionizing radiation, it turns out, also has amplification, but only at 1 level, and it's usually pretty modest, okay? So there's a difference there. And so this is, this is very interesting observation, and it's incredibly chilling because, of course, we're exposed to these damn things all the time. And so we're chewing up our DNA. And we're doing that both in the cells of our bodies and also at least in the sperm cells, because we know that these DNA changes occur in sperm following EMF exposures, okay? So, so we're producing, mutations that are passed on to our children as a consequence of this exposure. So all of the industry propaganda is just wrong. I mean, every single thing that they say. When you look at the actual science, it falls apart. Okay, now, now let me just say, and I'm not going to spend, what do we got here? We've been at it for quite a bit of time. I better hurry up here. Okay, so there are lots of things that we talked about, and all of these can be produced through that figure that I talked about before, the one that was on this figure basically explains each of these things. And I don't think I have time to go through these, but basically, so you got oxidative stress, you got single strand breaks in DNA, double strand breaks in DNA, 8-hydroxy-DG in the DNA, so that's an oxidized base in the DNA. Cancer, breakdown of the blood-brain barrier, male and female infertility, therapeutic effects, depression and other diverse neuropsychiatric effects, melatonin depletion and sleep disruption, cataract formation, and then the cardiac effects that we talked about before. All these things you can explain through that mechanism, okay? So the one thing we can say for certain is that when industry claims there can't possibly be a mechanism for each of these things, they're wrong, okay? And so that's where, there are also hormonal effects from both various hormones and also steroid hormones. They work differently. In fact, in some ways, they work oppositely. And then there's cell death. Apoptosis can be produced by both intracellular calcium elevation and also by DNA changes, including 8-hydroxy-deoxyguanosine. So anyway, so everything there can be explained, okay? So what I'm trying to tell you is there's no mystery anymore, okay? That that the industry claim that none of these things can occur, there can't possibly be a mechanism. It's total nonsense. Okay, now, so these are not the only pathophysiological effects that are going on, but they're the ones that are best understood, and and the ones where we have the best ideas about how they work. So, and so when you look at those effects, what do? They affect each of the 4 things I think we most value as individuals and as a species. They impact our health in many different ways. They impact our brain function severely often. They destroy the integrity of our genomes and our ability to produce healthy offspring. They're all impacted. So you wonder, and so that's pretty bad, obviously. It turns out when you look at some other things, it gets still worse. I hate to bring you all this bad news. Now, let me just say, I don't have time to talk about EHS, but basically there are reasonable mechanisms for understanding EHS as part of this VGCC thing, and I can't really spend the time on this. So I just want to tell you that my opinion is that the VGCC mechanism can explain a lot of the things about EHS, but we really don't have time to talk about that. So anyway, so I'm just going to skip through this. Okay. Okay, now let's get to the end of this. What I want to do now is talk about things I call worst case scenarios, and there are 5 of them. And basically 2 of these I think are already in the works and we see them and there shouldn't be any question about them. The other 3 are, you know, slightly questionable, but I think highly probable. That's my assessment. Maybe I'm wrong about 1 or 2 of them, but I doubt it, okay? And, you know, and so these are things which collectively can basically destroy us. So one thing that I think is probable, but perhaps not quite certain, is that the autism epidemic is probably caused by EMF exposures, that the EMF exposure is the main driver of the epidemic. And what's true is that if you look over here, if you look at that sequence that goes from the low intensity microwave frequency, microwave/lower frequency EMFs, right in the top, middle top, okay? And then you got arrows that are kind of angling down towards the lower left, okay? That I think is the main sequence of events that's occurring between the EMFs and autism, okay? And so in autism, the main things that are clearly central to the causation of autism is the disruption of the formation of the synapses in the developing brain, okay? The thing that makes autism unique is that. If you don't form, so the synapses are the connections between the neurons, okay? And they have to be at least approximately right or you're going to screw up the brain function in a variety of different ways. And the synapse formation involves these 5 mechanisms that are listed under it, dendritic outgrowth, synapse, etc. Those 5 things are all involved in the formation of proper synapses. In the developing brain, every single one of those is regulated by intracellular calcium. You got too much intracellular calcium, you're in deep trouble, okay? And so I think that's basically what's going on here in the autism epidemic. There are also other things that are going on, however, and something like 12 to 15% of the autism patients carry what are called de novo mutations. That is, mutations that were not present in either parent and that have a role in causing autism that affect the synapse formation, okay? About 12 to 15% of them. So not most of them, but a substantial minority of them have these mutations in them. And those mutations can be caused by the things that we just talked about that occur to the cellular DNA, okay? And I'm talking about specifically that those mutations can be caused by single-stranded DNA breaks, double-stranded DNA breaks, and oxidized bases in the DNA, okay? All you know, so the mutations that have been shown to be involved in these de novo mutations in autism have those properties. So I think those mutational events occur as well. What does that tell you? It tells you that the exposures in the parents are important, not just the exposures to the developing fetus and the baby after birth, okay? They're all important. Now, I'm not saying there are no chemical effects. I think there are chemical effects, but they also go through calcium. And that's basically what's shown going from the upper left down to the lower right, okay? So it turns out there are 32 kinds of 32 kinds of studies that document that main sequence going from low intensity microwave frequency EMFs down to the synapse formation that have been various kinds of studies that have been basically that whole pathway has been documented from 32 studies of autism, okay? And I've presented this now several times at the Autism 1 meetings. Yeah?
Just a question about that. So is it because the parents are exposed to the electromagnetic fields that the fetus gets created that way, or is it while the fetus is in
Well, okay, so what I was trying to say is that these de novo mutations have to occur in one of the parents before conception, okay? But then in addition, and so those are caused by exposures in the parents. And there is some evidence, by the way, for this, and particularly in the males, because more of the mutations are actually produced in the male parent than the female parent, okay? In general, in humans. And so, you know, when you see exposures in the father that influence the occurrence of that increase the occurrence of autism in the child, then that almost has to be explained in this way. But I think the main effect of these environmental exposures is in the developing fetus and shortly after birth, okay? And they then directly influence the synapse formation in the developing brain. Is that clear?
Yeah, it's scary.
It is scary. Almost everything I say is scary. I wake up with nightmares, you know. It's, we're in a horrible situation. We really are. Okay, so that's autism, okay? Now, these neuropsychiatric effects. Here's all these neuropsychiatric effects have been found in many different studies. And I mentioned some of these before. Sleep disturbance, insomnia, headache I didn't mention. Depression and depressive symptoms, fatigue and tiredness. Dysesthesia is basically that the sensory systems don't work properly. So you can have problems with vision, with hearing, and smell is olfactory. You get problems with concentration, attention, cognitive dysfunction. Memory changes, mainly I don't remember well, and dizziness, irritability, loss of appetite and body weight. You can also get increases in body weight. So, you know, so it can go either way depending on different conditions. Restlessness, tension, anxiety, stress, agitation, feeling of discomfort, nausea, and you also get skin tingling and burning and inflammation, and something called dermographism that I won't try to explain to you. So these are all skin things. And so, you know, you look at these things, so many of them, I mean, this is what everybody's complaining about, right? We know they're caused by EMF exposures, okay? And we know those DMF exposures are produced basically because of the massive effects on the brain. And we know that's produced because there are very high densities of VGCCs on every single neuron in the entire nervous system. Okay, so the whole nervous system is the most sensitive organ in the whole body to these EMFs. That doesn't mean there aren't lots of things going on elsewhere, but lots of stuff is going on here, that's for sure. And so basically we're going to understand this whole thing, and there have been extensive studies done on the effects in the brain in experimental animals exposed to these EMFs. And they're really stunning. I've spent hours going over those studies. Okay. Worst case scenario number 3, sterility and spontaneous abortion. Production goes to 0. Is that ridiculous? Maybe. Okay, so first of all, we know that, you know, that for instance, there are declines in sperm count and sperm quality all over the world. And you know, these things, so male fertility is much easier to study than female fertility. And there's evidence that you know, the male and female infertility in general are infertility is increasing, as well as spontaneous abortion. And we know these can be caused, all of them can be caused by microwave frequency EMFs. There was an interesting study that was published by Magras and Zinos almost 20 years ago now. And what they did was to take pairs of mice, so one male and one female, young, nice, young, healthy mice, and you put the two together in a cage on the ground, outside, in what's called an antenna park. So there are a bunch of broadcasting antennas there, broadcasting television or radio antennas, I think probably mostly television. And the exposures at ground level were well within our current safety guidelines. So if the safety guidelines have any relevance, there shouldn't be anything happening, right? Nothing should be happening. And they put the mice, these little cages in 2 different locations, one with a somewhat higher level exposure, another with a somewhat lower level exposure, but all, as I said, all well within our current safety guidelines. And what they found was, at the higher level exposure, the mice produced 1 litter that was fairly normal in size. So let me say, gestation period in mice is 30 days. They're real quick, okay? So you can this is an easy study to do, and you can do it very quickly. And so, you know, something like 30 days later, they produce a litter, and it's perhaps not quite normal, but roughly normal, okay, size. Then they produce a 2nd litter that was down quite a bit in numbers. And then complete sterility. Not a single offspring following that, okay? So they go along for a while and things aren't obvious, and then you start seeing they're obvious, but they're still producing, and then they go off the cliff. Reproductively.
Excuse me, question.
Yeah. Go ahead.
This antenna park doesn't sound like it has microwave and ELF coming out, but the frequencies that are entirely in between.
Yes, that's right.
So how does that prove what we're talking about?
Well, all of these things work via the VGCCs. The whole spectrum works via the VGCCs.
So
yeah.
all the frequencies are now implicated.
Yes. Yeah. Yeah. Well, that's you know, as I mentioned, the extremely low frequency one, some of the intermediate. These are intermediate frequencies, or what are probably more properly called radio frequencies. The industry often calls the microwave frequency stuff radio frequency. I don't argue with the terminology, but anyway, so this is really, so basically I think we've got a situation where we know there are reproductive effects of these exposures, where we know there are problems with particularly with male fertility that are getting more and more serious with time. And so the question is basically, how long does it take before the human population goes off the cliff? And my guess with these ever-increasing exposures is probably something like 5 years. I think we're going to have a huge decline in reproduction. And there, you know, that's my prediction. Now, am I certain about the timing? No, but that's you know, it seems to me that's a very good guess based on where we are now and the properties of this animal study. And so, you know, I really think that we're looking at this a fairly short time period. You know, the problem is if our reproduction goes off the cliff, we're going to be in very deep trouble from that alone. You know, basically, what's a society with no future, or almost no future? Okay, so worst case scenario number 4, huge numbers of germline mutations. Well, we've already said that, you know, that you can get huge amounts of free radicals from these EMF exposures because of the amplification mechanism. It's the free radicals that attack the DNA, and we know these occur in the male germline because they've been studied there quite a bit. And so, you know, you produce lots of mutations. It's been estimated in humans that all you have to do is increase the mutation frequency by about a factor 2.5 to 3, and we will become extinct. Why? Because we're producing so many damaging mutations that almost all the progeny have very serious problems. Okay, and that's so that's another thing that you know that can happen. And let me just say again, in the autism patients, we're seeing lots of mutations. but it's still a minority of the autism patients that have new mutations. But there are many, many other kinds of genes that can mutate that can cause serious problems. They're just not the same problems that you see in autism. So anyway, there are 3 studies in which they showed that, that EMF exposures increase mutation frequencies, and I've listed the references in this. So we may well be destroying our biological inheritance. So we took something like 3 billion years to develop our biological inheritance. We may be destroying it in a few years, the way this is going. So can you believe this? I mean, it's unbelievable, isn't it? Huh?
What is a germline?
Germline are the eggs and sperm, the things that produce the next generation. Okay? Yeah, germline's the eggs and sperm. And so, you know, as I say, these have been studied in sperm, not as much in the eggs because it's much harder to study. Even in animals, it's harder to study. So, yeah. Okay. So, number 5. Okay. You probably say I've had enough. We have now an epidemic, still a small one, of very, very early onset Alzheimer's. People age 30 coming down with Alzheimer's disease. This was unheard of in previous years. It's still rare, but it occurs. Okay. Now, why do I think that it might have something to do with these EMFs? Okay. So that's one of the you know, one of the things that shows that this is happening. Now we know that from occupational exposure studies, these are old ones actually, that exposure to high amounts of our power wiring EMFs will increase the occurrence of Alzheimer's disease. So people who, as part of their occupations, work a lot with power wiring at high densities tend to have higher amounts of Alzheimer's disease. And as I say, that works via the same mechanism, by the VGCC activation. So we don't have any comparable studies with the, you know, the microwave frequency EMFs, which is a whole, but, you know, lack of knowledge doesn't tell you it's not going on. There's lots of things we have lack of knowledge on because basically there's no research going on in the US in any of these areas. High levels of intracellular calcium have very important roles in Alzheimer's, and there are quite a number of reviews. I just put one of them in here that documents that, okay? So, you know, CO2 plus I, important for Alzheimer's. There's also a VGCC polymorphism in one of the VGCCs, the one that's, you know, that's one of the ones that's very important, and where one of the common forms of the gene, relatively common anyway, produces more of the channel. Right, so you get more channel from that form of the gene than from the other common form. And that form that produces more VGCC is associated with increased Alzheimer's. So it produces increased susceptibility in Alzheimer's. Okay? So that tells you the VGCCs can have a role in Alzheimer's. And there are 2 studies that came from a group by Zhang et al. And what they did, which was quite interesting, this is a rat study. They took rats and they exposed them to a series of very short pulses of microwave EMFs, and as very young rats. Okay, so young rats, and you give them a bunch of pulses, and then you stop. You don't give them any more pulses. And then you wait, you know, I don't know, I forget, 10, 12 months, until you got something that might be a middle-aged rat, and you ask, what's it doing? You know, what's different about it? And what you find is the middle-aged rat looks like it's got Alzheimer's disease. Why do I say that? It's got oxidative stress in the brain. It also has high levels of the amyloid beta protein, which is characteristic of Alzheimer's, in the brain of the rat. and then you also have cognitive and memory impairment. So if this is right, all you have to do is give a few a few pulses, or maybe more than a few pulses at a more or less young age, and you get Alzheimer's disease. So yeah, I mean, this is quite stunning. In fact, the pulses they use are at least superficially to a lot of the pulses you get off smart meters. Now, could it be the case that, that the smart meters are causing millions and millions and millions of cases of premature, very early onset Alzheimer's disease? We don't know. That's exactly the problem. We don't know. Why are we taking these kinds of incredibly stupid, why are we doing these incredibly stupid things with absolutely no sense of the kinds of things that can be produced through the EMFs? It's just, I mean, I just can't believe how stupid and destructive we are. It just totally blows my mind. Now, Alzheimer's disease. Okay, there's another study that. Okay, yeah. Anyway, Alzheimer's disease typically has a long latency period in humans. Okay, so in humans, typically, there are certain stressors that we know can sort of start the process that causes Alzheimer's disease, but you don't actually get the disease until something like 25 years later. Okay, so you start, you see the symptoms much later. So there's this long process, normally, that occurs during the latency period before you see overt symptoms of Alzheimer's, which raises an interesting question. The people age 30 coming down with Alzheimer's, they may be responding to the exposures that we had 25 years ago, when they were vastly lower than they are now. Okay? Now, if that's true, and I don't know that it's true, but it seems reasonable that it may be true, then even if we stopped all exposures tomorrow, completely stop exposures tomorrow, we may well have a huge epidemic of very early onset Alzheimer's disease coming down the pike over the next 25 years from the exposures we've already had, okay? And from these ever-increasing exposures that we've already had. So, how can we be so incredibly stupid? I mean, it's just incredible. How can we be so stupid? Okay, so, now let me just say, each of these, 5 case, even these 5 what I call worst case scenarios, are, I might be wrong about 1 or 2 of them. But even if I'm wrong about 3 of them, we're in very deep trouble. So let's just think about this. We're going to have a big crash in reproduction. I don't know exactly when it's going to occur. I think it's going to occur pretty quickly, but, it's going to occur. We're going to see a huge dip in reproduction. I think the neuropsychiatric effects are already occurring. We know they're caused by EMFs. They're all over the place. There's no question about it. That's already occurring. Okay. But you throw into that the fact, so we may have a 60, 80, 90% drop in reproduction. Many of the children born may well have autism. Many, okay, meaning it could be. So let me just say the last good figure we have on the autism incidence was based on children born in 2002 in the US, okay? And they were found to have an autism frequency of 1 in 68, okay? Now, here we are 15 years later. We don't have the foggiest idea what it's going to be in children born this year. It could easily be, given the increases we've been seeing, 1 in 20, 1 in 10, maybe even 1 in 5. I mean, so we may already be in an incredibly, stunningly difficult situation with autism alone. And of course, each autism child has a massive impact on the family, right? Okay. Then we've got germline mutations, which will cause many other problems with young children. They may be extremely common. And finally, we've got things like very early onset Alzheimer's disease, which may be all over the place, okay? Now, I don't know, yeah. Oh, we're late. Oh, we're way off, aren't we? I'm sorry. Well, I mean, I'm finished, okay? So I can't draw any more things, but do we have time for some questions?
Sure, if you folks want to stay, if anybody needs to leave, by all means, feel free.
Yeah. So why don't you come up and use the microphone? And I guess you have to, yeah. Yeah. Mm-hmm.
Okay. At one point, I read that you had done some work on helping people recover from electrical sensitivities. And you're suggesting some maybe calcium that I can't remember. I don't know what it was.
Okay.
Is there any hope for recovery from some of those issues?
Okay. So EHS, is there any help for recovery? I think there is, but I don't have enough knowledge to say for certain that there is. I think there are a number of things that can be useful with EHS, and they're actually quite similar to the same things that seem to help MCS, multiple chemical sensitivity people, probably because the mechanism is very similar. And those include, for one thing, magnesium supplements. We tend to be, our diets tend to be quite low in magnesium, partly because of the kind of foods we eat and partly because the soils, the agricultural soils, have gotten depleted in magnesium. So that's an issue. It turns out that low magnesium increases both the NMDA receptor activity, which is involved in the chemicals in MCS, but also the VGCCs. So when we are magnesium deprived, we tend to make more VGCCs for whatever reason in our cells. And so that's going to increase the responses there, okay? So that's something. But there's another thing which I think is, and let me just say I'm a PhD, not an MD, so nothing I say should be viewed as medical advice, okay? I hope you know that. I think that one of the ways that is promising is to raise the level of a very important regulatory system called NRF2, NRF number 2. I published a paper on NRF2 in 2015. You can get this paper off the PubMed database at no cost. So if you just search under my name, P-A-L-L, and initials M-L, and NRF2, you'll find it, and you can get it. And you can look at that. There are a lot of different health promoting factors that raise NRF2. So look at the paper and see what you think. And I think that may be quite helpful. But again, I don't know for certain. And again, this is not medical advice, okay? I'm not saying these are the only things you should think about, but these are some of the ones that I'm thinking about the most nowadays. You want to? No, if you can't be heard on the video here, so yeah.
So if we all took calcium channel blockers, would we be all set and protected?
Okay, very good question. I wish it were true. Okay, I mean the problem is the calcium channel, the VGCCs are so important for so many different things. So that if we block them all, we're basically, we can't do anything, and we're totally dysfunctional. So that's not a useful. Now, does that mean calcium channel blockers are not useful at all? At this point, I don't know. I think I have some hopes that they will be useful, but you can't block everything because we need them. Okay, so that's a very good question. Thank you.
I'm wondering if you talked about the calcium ions flowing into the cell. Has that been captured on video?
Actually, in a visual, you can use a light microscope and see it. Well, what you see, okay, so let me, you're asking in response to the EMFs. Is that right? You're asking? Okay. Okay. Okay, I think basically the answer is no, but it can be done. But nobody's done it yet. Look, what we know is, and I mentioned this before, but there is evidence for calcium influx into the cell from studies, not by that method, by other methods. And there's also evidence that you do get an increase in calcium signaling following EMF exposures. Okay, so those things fit in with that. And there's also evidence that you get increases in nitric oxide that I mentioned before. Okay, in fact, that whole figure, basically, that I showed you, has been documented as the various events in that figure following EMF exposure. Okay, so that's not hypothetical. I mean, we've got evidence for all three of the pathways that are involved there. So now that I've destroyed your whatever. Oh, thank you, thank you. I appreciate that. Yeah. Well, yeah.
I do have one quick question.
Okay, sure.
Given that you're just, it seems to me you're just about to, by the way, thank you very much for this evening. Okay. And for your work today. Mm-hmm. It seems to me that you're just about to launch into what I'm about to ask. Okay. Do you have any thoughts on hope?
Do I have any thoughts on hope? Yeah. Yeah, I mean, you know, if I were sitting around depressed in the dumps and you know, I'd probably give up and not worry about giving these talks. I think there's hope, but it's going to be a major uphill battle, and I think that's pretty obvious. Look, you've got this, you know, trillion-dollar set of industries that's working as hard as they can to destroy us, and putting out the propaganda to block any resistance on the part of people who know better. And you know, it's going to be a huge challenge. On the other hand, everything that we care about, and I think you go around this room and you ask everybody, what do I care about? Everything we care about is being attacked, and severely attacked by this. We have got to respond. We have got to take this and run with it. Because if we don't, we will have betrayed everything we care about. And that occurs for everybody in this room and everybody in the next room and so forth and so on, all the way around the world. So, you know, that's all I can say is we have to work together on this thing. Each of us has our own roles. You know, my role is to mainly focus on the science, but obviously that's not the only thing I'm doing. And, you know, that's what we got to do. So if there is hope, that's where it's going to lie. And so, you know, that's all I can say. You know, that's it. Yeah.
I hang my hat on hope every single day. And when I had the privilege of meeting Frank Clegg, who was the former president of Microsoft Canada, he said, given a nudge, the industry can absolutely figure out how to make biologically safe mobile technology. That's not going to happen. They've got a wonderful business model going on until they get that nudge. So I'm very privileged to have worked with Patricia Burke and many others in this room who have been trying to do the legwork to get the attention of our legislators to start protecting the public. And we had hearings on 2 bills today. 3 more bills will come up in the fall, and likely 2 more that had just been introduced this month. So start where you are. Start looking around to see what your personal exposures are. Think about your secondhand radiation when you're using a mobile device in active mode. What might you do differently so that you're not exposing the children around you or people with compromised health or the elderly? Start where you are. And there's a stack of cards there from Wireless Education. In 40 minutes, you can take an online course for less than the cost of a movie ticket. We can all get educated on this today and start spreading it out to those who care. You can write in testimony for these bills. You can call Dr. Bob Knorr at the Department of Public Health. We've been working together on a series of EMF fact sheets. If we can finally get a public authority to acknowledge this issue exists and offer some guidance, then maybe that will be enough for our schools to start paying attention in our municipalities, and so that we start holding the industry accountable and start repealing things like the Telecom Act of 1996 that basically says municipalities can't say whit about where they want a cell tower if they want one. In other countries, they will say, telecom company A, you can put a cell antenna here. Telecom company B, you can put one over here. In our Telecommunications Act, they built in a nice little clause for the industry that says, if you grant permission for one, you have to grant permission for all. So that's why when you go down Cedar Street and you look up at that water tower, count them. There's what, 30 or 40 antennas up there in that poor neighborhood with all those families right at the base of that cell tower, basically. So we all need to start being observant. We all need to start listening to our world experts and start taking action. I know I circled my tail on this for a little bit before I started taking action, but if not us, then who? So thank you. Thank you all for coming tonight.
Now, I just want to say one other thing. You know, this country takes pride in its science. And we do it in part because, you know, we've got more Nobel laureates than any other country by far, and so we think, hey, we're the top of the heap and we're doing all this great stuff, right? The fact of the matter is, in this area, we're doing essentially nothing. What happened was in 1986, the Environmental Protection Agency, which had an in-house part of the organization in-house that was doing research in this area, it was shut down. At essentially the same time, the Office of Naval Research, which had been funding quite a bit of research in this area, quit funding any new grants. A few years later, the NIH quit funding any new grants. So there's no government funding in this country. And for that reason, every single study that's been published on health effects of people living near cell phone towers comes from elsewhere. From that reason, there are, I think, 14 of them. They're all coming from other places. For that reason, each of the 19 studies on genuine Wi-Fi effects come from elsewhere. We're not doing diddly squat. And it's pretty much the same thing with regard to genuine cell phone exposures. You know, people use cell phones. You know, I mean, our science has been corrupted. And if this is the criterion of science corruption, we're the most corrupt country in the world. Now, I'm not saying that's the only criterion we should use, but you can see how serious the situation is as a consequence of the lack of study. And so for example, and you know, everything that we're not doing isn't being picked up elsewhere. For example, you know, there are 11 research groups that have argued that that the EMF exposures are at least a major cause of the autism epidemic. There hasn't been a single experimental study anywhere in the world to look at autism in children who were born from families living near cell phone towers, or you know, or from people who have Wi-Fi in their house. Of course, now almost everybody has it, so maybe that's not a useful study to do anymore. I mean, you know, so it's amazing, you know, the level of ignorance here is just stunning, and we're in really deep trouble because of that. So there's lots of things that need to be done, and I just hope that some of you will feel up to doing some of the things that need to be done, because it's essential for the survival of anything like humanity into the future. So thank you.